सो आज का बुक रीडिंग सेशन में हम जनरली बुक रीडिंग करने वाले हैं इंटीग्रेटेड टेक्स्ट बुक ऑफ बायोकेमिस्ट्री सेकंड एडिशन इंदुमती भी सौभाग्य लक्ष्मी ओके पारस मेडिकल पब्लिशर का ये बुक है। तो आज हम स्टार्ट करेंगे। वैसे इसको कई बार मैं पढ़ चुका हूँ, आज फिर से स्टार्ट कर देते हैं। चैप्टर हमारा 1 रहेगा, चैप्टर नंबर 1 सेल एंड ऑर्गेनेल्स ट्रांसपोर्ट अक्रॉस सेल मेम्ब्रेन। ठीक है, ये थोड़ा marker enzyme of a cell membrane and subcellular organelles. Since each organelle has a unique morphological feature, the purity of a organelle preparation can be assessed by examination in an electron microscope. Alternatively, organ-specific enzyme marker can be used to identify subcellular organelle. For example, the enzyme acid phosphatase that cleaves terminal phosphate group from substrate and has a pH optimum in the acidic range is localized in lysozymes, while enzyme succinate dehydrogenase is localized in mitochondria. By monitoring where each enzyme activity is found during a cell fractionation protocol, one can monitor the fractionation of lysosome and mitochondria respectively. Marker enzyme also provide information about contamination and biochemical purity of the fractionated organelles. The protein cytochrome C is present only in mitochondria, so the presence of this protein in a fraction of lysosome would indicate its contamination by mitochondria. The specific marker enzyme to identify individual organelle of the cell are shown in table 1.2. So different organelles and their specific marker enzyme. Subcellular organelles marker enzyme. प्लाज्मा मेम्ब्रेन को आइडेंटिफाई करने के लिए 5' न्यूक्लियोटिडेज एडेनिल साइक्लेज एंड Na+K+ एटीपेज। साइटोप्लाज्म के लिए मार्कर एंजाइम है लैक्टेट डिहाइड्रोजनेज। न्यूक्लियस के लिए DNA पॉलीमरेज, RN पॉलीमरेज, लामिन A एंड C। देन माइक्रोजोम जो कि एंडोप्लाज्मिक रेटिकुलम के लिए ग्लूकोज 6 फॉस्फेटेजेस, अल्फा ग्लूकोसिडेज। गोल्गी कॉम्प्लेक्स के लिए गैलेक्टोजिल ट्रांसफरेज। माइटोकॉन्ड्रिया की इनर मेम्ब्रेन, आउटर मेम्ब्रेन। मैट्रिक्स के लिए एटीपी सिंथेज, सॉक्सिनेट डिहाइड्रोजनेज, साइटोक्रोम सी, मोनोअमाइनो ऑक्सीडेजेस और साइट्रेट सिंथेज। लाइसोसोम के लिए कैथेप्सिन, एसिड फॉस्फेटेजेस। पेरोक्सिसोम के लिए कैटालेजेस। Subcellular organelles. The cellular component are called subcellular organelles. They coordinate efficiently for the normal functioning of the cell. They are classified into 3 categories based on the presence or absence of a membrane. Organelle without membrane, ribosome and cytoskeleton are non-membrane bound organelle found both in prokaryotic cell and eukaryotic cell. 5, 6. ବସିଲା ଶୀତ ଦିନ ଶଳା ମାଡ଼ିଲା ଥଣ୍ଡା। single membrane bound organelles, vacuole, lysosomes, Golgi apparatus, endoplasmic reticulum are single membrane bound organelle present only in eukaryotic cell. Double membrane bound organelle, mitochondria nucleus are double membrane bound organelle present in a only in a यूकैरियोटिक सेल. सो सिंगल मेम्ब्रेन बाउंड ऑर्गेनेल वैक्यूल्स हो गया, लाइसोसोम हो गया, गोल्गी apparatus, एंडोप्लाज्मिक रेटिकुलम आर सिंगल मेम्ब्रेन बाउंड ऑर्गेनेल प्रेजेंट ओनली इन यूकैरियोटिक सेल, दोनों यूकैरियोटिक. ओके. एंडोप्लाज्मिक रेटिकुलम. नाउ endoplasmic reticulum is a single membrane bound organelle found in all eukaryotic cell. It constitutes 30 से लेकर के 60% of the cell that extend through the cytoplasm connecting cell membrane to the nuclear membrane. It is an interconnected network of a flattened membrane enclosed sac or tubular structure known as cistern filled with fluid that is different from the fluid in the cytosols. Figure 1.7. During cell fractionation, the endoplasmic reticulum is disrupted and automatically reassembles to form microsomes. There are two different types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. ये न्यूक्लियर एनवलप होता है, ये न्यूक्लियर पोर. स्मूथ एंडोप्लाज्मिक रेटिकुलम में आपका जनरली ग्रैन्युल्स नहीं पाएगा, रॉफ में ग्रैन्युल्स पाएगा. दीज आर राइबोसोम्स, दीज आर सिस्टर्न फोल्डिंग. देन ये आपका सिस्टर्नल स्पेस. ओके, बीच में जो स्पेस होता है सिस्टर्नल स्पेस, दीज ऑल आर सिस्टर्न. दिस इज राइबोसोम, दिस इज न्यूक्लियर pore, this is nuclear envelope, everything correct. Now rough endoplasmic reticulum. Now it is a figure of structure of endoplasmic reticulum. एंडोप्लाज्मिक रेटिकुलम यहाँ पर पढ़ा था एंड यहाँ पर ये रफ. सो रफ में आपको देखना राइबोसोम देखने को मिलेगा, जबकि स्मूथ में कोई राइबोसोम देखने को नहीं मिल रहा है. देन रॉफ एंडोप्लाज्मिक रेटिकुलम. ये दो प्रकार का है, एंडोप्लाज्मिक रेटिकुलम, रॉफ एंडोप्लाज्मिक. ओके. Then rough endoplasmic reticulum. Rough endoplasmic reticulum has a dense granular ribosome lining its surface, giving the rough surface. It lies immediately adjacent to the cell nucleus and its membrane is continuous with the outer membrane of a nuclear envelope. The ribosome on the rough endoplasmic reticulum are the site of a protein biosynthesis. The signal sequence on the protein synthesized direct them into the endoplasmic reticulum for processing. Proteins synthesized by the rough endoplasmic reticulum have a specific final destination. Some proteins, for example, remain within the endoplasmic reticulum, whereas others are sent to the Golgi apparatus, which lies next to the endoplasmic reticulum. From the Golgi apparatus, the protein are directed to lysosomes or to the cell membrane and secreted to the cell exterior. ये थंडा होता है छाड़ी हत्या करदेलानी. Smooth endoplasmic reticulum. Smooth endoplasmic reticulum SER is smooth in appearance without the ribosome lining. It acts as a storage organelle. It is involved in the synthesis of lipid, including cholesterol, phospholipid, which are used in the production of a new cellular membrane. New cellular membrane. In certain cell types, smooth endoplasmic reticulum plays an important role in the synthesis of steroid hormone from cholesterol. It supplies calcium for cellular function. In cells of the liver, it contributes to the detoxification of drugs and harmful chemicals. Microsomal cytochrome P450 hydroxylate drugs such as phenobarbital, morphine, aniline, aminopyrin, benzopyrin, etc. Ribosomes. Ribosomes are a small granular structure made up of two-thirds of the RNA and one-third of the protein. They are non-membrane bound and important cytoplasmic organelle found in close association with the endoplasmic reticulum to form rough endoplasmic reticulum where protein synthesis takes place. Free ribosomes are involved in the synthesis of intracellular proteins. They are named as 70S saved Borg unit found in prokaryotes and 80S found in eukaryotes. The letter S refers to the density and the size and known as saved Borg unity. They are composed of a two subunit, small and large subunit. 70S has 30 plus 50S subunit, 80S has 40S, 60S subunit. The two subunit are joined to each other by interaction between rRNA in one subunit and a protein in another subunit. Small ribosomal subunit read the messenger RNA and large ribosomal subunit form polypeptide chain from amino acid. Figure 1.8. Now large subunit, small subunit, large ribosomal subunit, E site, P site, A site, small ribosomal subunit, mRNA binding site. Okay. Structure of ribosomal subunit. Okay. Golgi apparatus. Hey Bhagwan. The Golgi apparatus was observed in 1897 by Italian cytologist Camillo Golgi. In most eukaryotic cell, Golgi body is present in all cell except RBC. It is located in the cytoplasm next to the endoplasmic reticulum and near the cell nucleus. It is a single membrane bound organelle, has 8 से लेकर के 10 broad flattened membranous stacked pouches called cisternae. They are arranged in parallel semicircular array held together by matrix protein, and the whole of the Golgi apparatus is supported by cytoplasmic microtubules. It has three primary compartments known as cis: cisternae nearest to the endoplasmic reticulum, medial central layer of cisternae, and trans-cisternae farthest from the endoplasmic reticulum. Function: carbohydrates are added to the nascent protein when they are moving through the endoplasmic reticulum, form cluster of fused vesicles. ये कितना नंबर पे 6 नंबर हो गया 7. स्ट्रक्चर ऑफ गोल्गी apparatus. ओके ये हो गया secretory vesicle, न्यूली फॉर्मिंग वेसिकल, दिस वन. स्ट्रक्चर ऑफ गोल्गी apparatus ये तो हो गया. ये सीस फेज, ये आपका ट्रांस फेज, इनकमिंग ट्रांसपोर्ट वेसिकल. ओके ठीक है, ये आपका लुमेन है. दिस एरिया इज योर लुमेन. ओके दिज आर सिस्टर्न. और क्या, ये न्यूली फॉर्मिंग वेसिकल है, ये सिक्रेटरी वेसिकल. क्लियर. यहाँ तक हमने देखा है. फंक्शन हमारा ये है 6 पेज 7. These fused vesicles migrate along the microtubules through a special trafficking compartment called vesicular tubular cluster that lies between the endoplasmic reticulum and the Golgi apparatus. When a vesicle cluster fuses with the cis membrane, the contents are delivered into the lumen of the cis phase cisterna. As protein and lipid progress from the cis phase to the trans phase, enzyme within cisternae modify macromolecules by glycosylation, sulfation, and phosphorylation. They are sorted, packaged into vesicles with particular labels for delivery to specific intracellular or extracellular location. Then mitochondria. Mitochondria are called the powerhouse of the cell because they are the site of aerobic respiration in the cell, where energy released from oxidation of foodstuff is trapped as chemical energy in the form of ATP. It is a double membrane bound sausage shaped organelles found in all eukaryotic cells. A cell consists of around 2000 mitochondria, occupying about one fifth of the total cell volume. They usually vary in their size, having a diameter between 0.5 से लेकर के 1 micrometer diameter and length up to 7 micrometer. RBC do not contain mitochondria, whereas the tail of the spermatozoa is fully packed with mitochondria. The double membrane divides its lumen into two distinct aqueous compartments. The inner compartment is called matrix, which is folded into cristae, whereas the outer membrane forms a continuous boundary with the cytoplasm. Figure 1.10. Mitochondria have their own circular DNA, RNA molecules, ribosome, the 70S, and few other molecules that helps in protein synthesis. Mitochondrial DNA is a double-stranded, highly coiled circular DNA. It has about 16,569 nucleotides. About 10% of a mitochondrial protein are produced in mitochondria. The mitochondrial protein encoded by nuclear DNA are synthesized in the cytoplasm and translocated into mitochondria by TOM, मतलब translocase of outer membrane, and TIM, मतलब translocase of inner membrane protein complex function. The most prominent role of a mitochondria is to produce the energy currency of the cell, ATP, through electron transport chain and oxidative phosphorylation. भगवान स्ट्रक्चर ऑफ माइटोकॉन्ड्रिया. ये आपका डीएनए है. ये वाला पोर्शन क्या है? ओके, इसको विजुअलाइज किया गया है. दिस इज F0 पोर्शन. ये बड़ा है F1 पोर्शन. ओके, डीएनए, राइबोसोम, मैट्रिक्स आपका ये हो गया. आउटर मेम्ब्रेन दिस वन, इनर मेम्ब्रेन आपका ये है, मतलब मैट्रिक्स वाला एरिया. ये आपका इंटर मेम्ब्रेन स्पेस, ये वाला, ये वाला, ये होगा आपका आउटर मेम्ब्रेन. ठीक है, ये आउटर मेम्ब्रेन, ये इंटर मेम्ब्रेन स्पेस, ये वाला, इनर मेम्ब्रेन, इसके बीच मैट्रिक्स हो गया ये. And these are a crystal. Structure of a mitochondria, F0 portion, F1 portion. Okay. TCA cycle, oxidation of a fatty acid, few steps of a urea cycle, and heme synthesis takes place in the matrix of a mitochondria. The mtDNA has information for synthesis of a 2 rRNA, 22 tRNA, and 13 protein, which are component of a electron transport chain, etc. The inner mitochondrial membrane contains cytochrome P450, which is involved in steroidogenesis. The antioxidant enzyme superoxide dismutase, that is superoxide dismutase, is present in mitochondria. Then coming to the lysosomes. Lysosomes are single membrane bound subcellular organelles. They are spherical vesicles that contain hydrolytic enzyme that can break down many kinds of biomolecules. The lysosomal enzyme act only locally, but if the lysosomal membrane is disrupted, the enzyme can cause autolysis of a cell constituent, hence known as suicide bags. The pH inside lysosome is acidic, मतलब pH 5, and most enzymes are acid hydrolases. The lysozymes contain around 40 enzymes which digest and remove the unwanted metabolic waste substances. The hydrolyzed products are utilized by the cell. Enzyme of the lysozymes synthesized in the rough endoplasmic reticulum are transported to the Golgi apparatus in small vesicles, and they are specifically tagged with mannose 6 phosphate so that they are properly sorted and transported to lysosomes. Synthesis of a lysosomal enzyme is controlled by nuclear genes. Mutation in the gene for this enzyme are responsible for more than 30 different human genetic disorders, which are collectively known as lysosomal storage disease. क्या ଲାଗିଲାଣି ଆଉ ପଢିପାରିବିନି. Lysosome act as the waste disposal system of the cell by digesting worn out organelle food particles through autophagy and engulfed viruses or bacteria by endocytosis. Endocytic vesicle and phagosomes fuse with lysosomes primary to form secondary lysosome or digestive vacuoles. Lysosomes can help to self-destruct in a process called programmed cell death or apoptosis. Then coming to the peroxisome. Peroxisomes are tiny spherical membrane bound organelles with granular matrix occurring in the cytoplasm of the eukaryotic cell. 7 number वाला 8 बेसिक बायो. ओके, देन लाइसोसोमल एंजाइम. ओके, यहाँ पर पेरोक्सिसोम के बारे में कुछ पढ़ रहे थे. ओके, ठीक है. लाइसोसोमल एंजाइम हमने कवर नहीं किया. ओके, लाइसोसोम को पहले कवर कर लेते हैं. लाइसोसोमल एंजाइम. सो टाइप ऑफ लाइसोसोमल एंजाइम एग्जांपल. सो फॉस्फाटेजेस में आपका एसिड फॉस्फाटेजेस आएगा, एसिड फॉस्फोडाइस्टरेज आएगा. ग्लाइकोसिडेज में बीटा ग्लूकोसिडेज आएगा, बीटा ग्लूक्यूरोनिडेज, बीटा गैलेक्टोसिडेज, अल्फा ग्लूकोसिडेज, अल्फा मानोसिडेज, अल्फा फ्यूकोसिडेज, बीटा एन एसिटाइल ग्लूकोसामिनिडेज, हायालुरोनिडेज, अराइल सल्फाटेज, लाइसोजाइम्स. देन प्रोटीएजेस के अंदर कैथेप्सिन ए, कैथेप्सिन बी, कोलाजिनेज, पेप्टिडेज, इलास्टेज। लाइपेज में एसिड लाइपेज, फॉस्फोलाइपेज ए, फैटी एसाइल एस्टरेज, फॉस्फाटिडिक एसिड फॉस्फाटेजेस। देन न्यूक्लिएजेस में एसिड राइबोन्यूक्लिएज, एसिड डीऑक्सी राइबोन्यूक्लिएज, फॉस्फाटिडिक एसिड फॉस्फाटेजेस। ओके, ये दो है या एक है, देखेंगे इस पर। देन यहाँ तक हमने पेरोक्सिज्म खत्म कर दिया था। They are synthesized from rough endoplasmic reticulum. They are prominently present in leukocyte and platelets. Function. Hey, Prabhu. Peroxisome are involved in the catabolism of a very long chain fatty acid, branched chain fatty acid, bile acid in intermediates. They also contribute to the biosynthesis of a membrane lipids known as plasmalogens. They transfer hydrogen from compounds to oxygen to create hydrogen peroxide H2O2 and then convert hydrogen peroxide into water. They contain peroxidase catalase enzyme with an important role in detoxification. Catalases break down highly toxic hydrogen peroxide to water and oxygen. Peroxisome of liver detoxify alcohol and poison by oxidation. Okay. Peroxisomal disorder are a group of hereditary metabolic disorder that occur when peroxisomes are missing or do not function correctly in the body. Then centrosome and centriole. Centrosome, Latin centrum, center, Greek जो ओके लैटिन टर्म जो होता है सेंट्रम, इसका मतलब सेंटर, एंड ग्रीक सोमा मतलब बॉडी, is an organelles that serve as the main microtubules organizing center, that is microtubule organizing center of the animal cell as well as a regulator of a cell cycle progression. They are found within the cytoplasm of the cell close to the nucleus and are involved in the cell division. Centrosomes are composed of two centrioles, a daughter centrioles and a mother centrioles, linked together by interconnecting fiber. They are arranged at right angle to each right angle to each other and surrounded by amorphous mass of a protein termed the pericentriolar material, that is PCM. Each centriole is a cylindrical array of 9 microtubules. The cell cycle consists of a G1, S, G2 mitotic phase. Mitotic phase include prophase, metaphase, anaphase, telophase. The centrosomes replicates once during the S phase of the cell cycle. During the prophase of mitosis, the centrosome migrates to opposite pole of the cells. During metaphase, the mitotic spindles are formed between the two centrosomes. Figure 1.11. These spindle fiber helps to separate the chromosomes. Upon division, each daughter cell receive one centrosomes. Aberrant Aberrant number of centrosomes in a cell have been associated with cancer. Okay, role of centrosome during cell division. Okay, ठीक है. Centrosome is an organelle that contain two centriole. Okay. Centrosome is an organelle that contain two centriole, whereas centromere is a highly constricted region on the chromosome. A centrosome is the microtubule organizing center, whereas centromere holds together the sister chromatid in a replicated chromosomes. Then nucleus. Nucleus, the storehouse of the cell's DNA, is a double membrane organelle, spherical in shape, located at the center of the cell in all eukaryotic cell except for mature RBC in the circulation. However, in some cell like small lymphocytes and spermatozoa, it occupies most of the available space. It comprises nuclear membrane, nucleolus, nuclear matrix, nuclear lamina, and chromatins. It is surrounded by nuclear membrane enveloping nucleoplasm. Figure 1.12. This membrane is double layer, an inner and outer membrane arranged parallel to one another and separated by 10 से लेकर के 50 नैनोमीटर. Outer nuclear membrane is continuous with the membrane of endoplasmic reticulum. The inner membrane, called as perinuclear membrane, has numerous pores called as nuclear spore. Nuclear pore are made up of multiple protein called as nucleoprotein. The pores are 100 nanometer in total diameter. However, the gap through which molecule freely diffuse is only about 9 नैनोमीटर वाइड ड्यू टू द प्रेजेंस ऑफ. ये 8 नंबर का पेज है. ओके, ये सेम है, ऊपर है. ये 9 नंबर का पेज. ओके. So, regulatory system within the center of the pore. This size selectively allows the passage of a small water-soluble molecule while preventing larger molecules such as nucleic acid and larger protein inappropriately entering and exiting the nucleus. Selective transport of regulatory molecule, enzyme, mRNA, rRNA, tRNA occurs through nuclear pores. Nucleus contain nucleoplasm, also known as karyoplasm, a gel-like around substance that contains genetic material DNA in combination with protein histone. It comprises enzymes which are involved in the synthesis of DNA, that is DNA replication, and RNA transcription. Within the nucleus, there are tiny spherical dense bodies called as nucleolus where ribosomes are assembled. The nuclear lamina is a dense fibrillar network associated with inner face of a inner nuclear membrane. It is composed of intermediate filament called as lamin ABC and lamin associated membrane protein. Along with mechanical support, nuclear lamina regulates DNA replication, cell division, chromatin condensation to chromosomes during mitosis. Nucleus is responsible for cell division, growth, differentiation. It controls main cellular metabolism through controlling synthesis of particular enzymes, allow level of gene regulation, and control gene expression. Vacuoles. Vacuoles is mostly defined as a storage bubble in the cell. These vacuoles might store food or any variety of nutrients a cell might need to survive. Vacuoles can store waste product so that the rest of the cell is protected from contamination, although eventually those waste products are sent out of the cell. Cytoplasm. Cytoplasm is a fluid known as cytosol jelly like substances present inside the cell extending from the cell membrane to the nucleus within the cell. It is mainly composed of water along with protein, carbohydrate, lipid, electrolytes. Cell organelles are embedded in the cytoplasm. These cell organelles contain enzymes mainly responsible for controlling all metabolic activities and chemical reaction within the cell. Cytosol is the site of a glycolysis HMP pathway, de novo synthesis of a fatty acid. साइटोप्लाज्म खत्म हो गया। उसके बाद अब क्या क्या बचा? द फंक्शन ऑफ वेरियस सब सेलुलर ऑर्गेनेल आर शोन इन टेबल 1.4। फंक्शन ऑफ सब सेलुलर ऑर्गेनेल, ओके, इसको खत्म करेंगे। हे भगवान। Function of various subcellular organelle are shown in the table 1.4. Function of subcellular organelle, subcellular organ function, ओके। रफ एंडोप्लाज्मिक रेटिकुलम का काम क्या है? साइट ऑफ प्रोटीन सिंथेसिस। स्मूथ एंडोप्लाज्मिक रेटिकुलम का काम क्या है? सिंथेसिस ऑफ कोलेस्ट्रॉल, स्टेरॉयड हार्मोन, फॉस्फोलिपिड्स, डिटॉक्सिफिकेशन ऑफ ड्रग एंड केमिकल। साइटोक्रोम P450। राइबोसोम का पार्ट ऑफ रफ एंडोप्लाज्मिक रेटिकुलम। रफ एंडोप्लाज्मिक रेटिकुलम का पार्ट होता है राइबोसोम, ओके। पार्ट ऑफ रफ एंडोप्लाज्मिक रेटिकुलम। साइट ऑफ प्रोटीन सिंथेसिस, गोल्गी apparatus, प्रोटीन sorting, पैकेजिंग, सिक्रेशन, ओके। माइटोकॉन्ड्रिया एटीपी प्रोडक्शन, साइट फॉर टीसीए साइकिल, ऑक्सीडेशन ऑफ फैटी एसिड, फ्यू स्टेप्स ऑफ ग्लूकोनियोजेनेसिस, यूरिया साइकिल्स एंड हीम सिंथेसिस, ओके। Lysosomes contain hydrolytic enzyme, digest macromolecules, foreign substance and worn out organelles. Lysosome, okay. Peroxisomes, catabolism of long chains, branched chain fatty acid, synthesis of plasmalogens, contain antioxidant enzyme, peroxidase and catalase. Centrosomes helps in the formation of a spindle fiber that separate the chromosomes during cell division, that is mitosis. Nucleus, control center of the cell, contain genetic material DNA, site for DNA replication and transcription. Nucleolus, site of a ribosome assembly. Nucleolus, site of a ribosome assembly. Vacuoles, storage of waste and other material. Cytoplasm, site for glycolysis, HMP shunt pathway, fatty acid synthesis, few steps of gluconeogenesis, urea cycles, हीम सिंथेसिस। देन सेल मेम्ब्रेन, अच्छा इसी को भी खत्म कर देते हैं। द सेल मेम्ब्रेन ऑल्सो नोन एज प्लाज्मा मेम्ब्रेन, दैट इज पीएम, और साइटोप्लाज्मिक मेम्ब्रेन एंड हिस्टोरिकली रेफर्ड टू द प्लाज्मा लीमा is a lipid bilayer that separates the interior of the cell from the outside environment, जो कि आपका एक्स्ट्रा सेलुलर स्पेस होता है। प्लाज्मा मेम्ब्रेन रेंजेस फ्रॉम 5 से लेकर के 10 नैनोमीटर इन थिकनेस। इन कंपैरिजन ह्यूमन रेड ब्लड सेल visible under light microscopy are approximately 8 micrometer wide and approximately 1000 times wider than plasma membrane. Function: cell membrane protects the intracellular component from extracellular environment. It acts as a barrier separating extracellular fluid and intracellular fluid. It is responsible for the maintenance of shape and size of the cell. So आज के स्टडी विथ मी में इतना ही, तो यहाँ पर हम इसे एंड करेंगे। आज का तारीख है 9/1/26। थैंक्स फॉर वॉचिंग।